We will now go for the last speaker from this part of the symposium. Our next speaker is Dr. Curtis Huttenhower from Harvard T.H. Chan School of Public Health in Harvard University, USA, with the talk, A Multi-Omic View of the Human Microbiome's Unique Molecular Activity. Please, Dr. Huttenhower, go ahead.
Great. Thank you so much. Thanks everyone for joining. Hopefully I am both audible and my slides are visible. Great.
Yes, yes.
They
All
are.
right. Thank you so much. I warned Maria Gloria already that we do have contractors here today, so I will hope that there are no unexpected interruptions. But if you see my house fall down in the background, that's why. In the meantime, I'm happy to join and talk a little bit about work that we do in the lab for ways to zoom in from big human population studies of the microbiome down to starting to get an idea of which microbial members, microbial strains, and sometimes individual genes or molecules are responsible for interesting microbiome-associated phenotypes. So this falls under the umbrella of a lot of work that our group does and the Microbiome Center at the School of Public Health does, that I'll mention at the end, where the goal really is to do exactly that, to start from large human populations that have been often studied for other epidemiology reasons, take some set of molecular measures of the microbiome in these populations, whether it's just metagenomic sequencing or metagenomics coupled with other types of more functional measures, compare these individual measurements to exposures and covariates that the populations have been experiencing, and then use existing, mostly good methods now to boil these molecular measures down to biologically informative features, do statistics on them to identify associations between microbiome features and host exposures or phenotypes. In some cases, for some phenotypes, there have been enough measures now that we can compare between studies, say for colorectal cancer, for example, to find consistent associations between the microbiome and a phenotype or exposure. And then really the goal here, again, is to start from these big population studies with lots of multi-omic measures and narrow them down to a few putatively causal bugs or pathways that can be taken out of messy human populations into a controlled lab environment, whether that's in mice or in vitro, for more mechanistic validation. One of the platforms that we have for doing that is the Human Microbiome Bioactives Resource, or HMBR, that's been developed as an NIDDK-funded resource over the past several years. This is a collaboration between our group, Owen White and Anoop Maharkar at the University of Maryland, Wendy Garrett, my co-director at the Microbiome Center at Harvard, and Ramnik Xavier at the Broad, where the goal of the HMBR is to identify bioactives from the microbiome, which we define pretty broadly, either as whole microbial members of the gut or other microbiomes that are responsible for a response to a phenotype or inducing a phenotype, either microbially produced small molecules, so things like protein products that are made by bugs that affect a phenotype, or more indirect mechanisms of microbial chemistry, say microbial modifications of dietary nutrients or pharmaceuticals that then result in a phenotype. So the goal of the HMBR is to be a resource. There are a variety of both data and software resources online for the web portal, in addition to things like protocols, results that we have from our own experiments. And this again includes everything from very upstream protocols for sampling and generating multi-omic data from large human populations. If you have a desire to collect tens of thousands of stool samples, feel free to contact us. We have a platform that makes that easy and cost-effective to do it at home now. We provide both pre-baked data resources with a specific focus on inflammatory bowel diseases and meta-analysis methods that allow combinations of data sets to be generated for other phenotypes. I'll speak today mainly about some of the computational methods that help to identify and prioritize different types of bioactive elements in the gut microbiome. And then Ramnik and Wendy, respectively, have worked on and shared protocols for both in vitro and gnotobiotic mouse-based validations and downstream investigation of putative bioactive elements. And credit to Owen's team for putting all of this together on the HMBR web portal. So some of the methods that we've looked at for prioritizing different types of bioactives, again, work a little bit differently, whether we're looking at bugs versus microbial gene products versus microbial chemistry. One of the methods that we wrapped up recently was led by a postdoc, now research associate, Yensong Zhang, which put together a system for specifically prioritizing proteins, specific proteins generated by individual microbes that are bioactive in terms of responding to or driving a phenotype of interest. The workflow is called MetaVivola, which is this complicated acronym, also a German cookie from what I understand. And how it works is by starting from assembled metagenomes so that we have the ability to find really novel, previously uncharacterized gene products that might be associated with phenotypes. MetaVivola will catalog the open reading frames from a collection of assembled metagenomes, first group those into nucleotide-based gene families, then cluster those gene families by amino acid homology into protein families. It categorizes protein families based on either strong enough homology to known proteins that we have a good guess of what they might be doing, remote homology to known proteins, so we recognize the protein family but can't necessarily assign it function via sequence, and then very low homology to anything that's previously been encountered. Any of these proteins, even the more remote homology, can be assigned some annotations based on secondary structural features, taxonomic profiles, phylogenetic profiling, and then either the characterized or uncharacterized protein families are prioritized with respect to a phenotype of interest based on these sequence features, ecological properties like how they are distributed among the population. Are they prevalent? Are they abundant? And then most important phenotypic properties, statistics of whether they associate with your phenotype of interest. These properties are all combined to give an overall ranking of which protein families might be bioactive in your phenotype of interest. One of the reasons this is so important is that even in the well-studied human gut, let alone less studied environments like developing populations, most protein families are uncharacterized. If we use the Human Microbiome Project's second set of data as an example, these are drawn from either inflammatory bowel disease patients or non-IBD controls. They're longitudinal samples. We have about 1,600 metagenomes from 130 participants divided between Crohn's disease, ulcerative colitis, and control individuals. Even among, again, this well-studied population, we start with about 45 million complete open reading frames. These cluster down to just over 2 million nucleotide gene families and about 1.6 million protein families, the majority of which are uncharacterized, either because sometimes they have very high homology to proteins that are themselves known but uncharacterized, or they have only modest or very low homology to any previously observed proteins. And for these, we're using the entirety of UniProt as a reference. So about 500,000 of these 1.6 million protein families have high enough homology to sort of believe to any protein that has any functional annotation. We're very generous about what we call a functional annotation here. The remaining 70% or so, even in a westernized human gut population, are uncharacterized. These can then be, even when lacking a current functional assignment, can be prioritized with respect to a phenotype like inflammatory bowel disease to identify protein families that might be responsible for or involved in the phenotype. So it turned out that we prioritized a surprisingly large set of chaperone usher-pillins, mostly types 1 and P pillins, that occurred mostly among Enterobacteriaceae. And we were, oh, there we go. We were able to confirm that these are differentially immunogenic. using sort of a natural experiment. These are so variable among different E. coli strains that we didn't even have to do genetics. We could take E. coli strains off the shelf that either carried or did not carry the operons containing these prioritized pilus proteins, which then turned out, in collaboration with Wendy, to be differentially immunogenic in MIT88 deficient cell lines to avoid a flagellin response. So what we think here is that different, slightly different pillin structures are more or less immunogenic, and having sort of the wrong strain of E. coli or Enterobacteriaceae can be one of the factors that sustains inflammation in IBD. We had a second sort of unrelated validation where we looked at a collection of prioritized von Willebrand factor homologs that were carried by different Bacteroides rather than different Enterobacteriaceae. And these are in humans, these are clotting proteins. So they're essentially Velcro proteins that help cells together. Our hypothesis was that these would be involved somehow in Bacteroides biofilm formation or mucus adhesion. And it turned out to be both. In a Bacteroides knockout strain, biofilm formation was disrupted in the knockout only in the presence of mucin. So we don't know any additional details yet, but these family of von Willebrand factor homologs are helping Bacteroides to aggregate specifically when adjacent to the mucus layer in the colon. So this work on metavibola and prioritization of bioactive protein families in IBD and other phenotypes is already published. I want to squeeze into the time today a little bit of description of some of the follow-up work we've been doing for other types of bioactivity around microbial chemical activity rather than protein products. This is work that was led by also postdoc, now research scientist, Emrisha Bosley. Emrisha put together a system kind of in parallel with Yensong and Metavibola that uses a similar intuition for chemical families rather than protein families. Again, the vast majority of chemical families that we encounter in the human gut are, in this case, not uncharacterized, but unannotated. So if you look at mass spec metabolomics of stool or the human gut, typically 500 to 1,000 unique chemical features are identifiable, and the rest are known to be unique, but can't necessarily be assigned an ID, just like an unidentified OTU, for example, or ASV. So one of the tricks we can use to get more information about these unidentified, unannotated chemicals is guilt by association. So this has been used for protein function assignment. It's been used for certain types of mass spectrometry. So for double mass spec, where we have spectral similarity, systems like GNPS use similarity among fragmentation spectra to identify related chemicals. We and many other studies don't always have double MS spectra. So we used a similar intuition again to look at covariant abundances of proteins, or excuse me, of chemicals for guilt by association, in addition to some other prioritization features drawn from Metavibola. So again, applying this system called macaron to the HMP2 data, here drawn from a subset of the same samples,
Minutes
we have,
30.
great, thank you. We have not quite 550 fecal metabolomes. These contain not quite 500 identified features and just under 45,000 total features. We can cluster all of these chemical features into co-abundance modules by covariation in abundance across the different microbiomes. And we've tuned macarons such that these modules are about the size that you would expect for a single chemical pathway. Of the 600 or so total modules, 150 of them from this particular data set include at least one of these annotated features, which we can then use to transfer information about its chemistry to other members of that module. In addition to this biochemical information, we can prioritize potentially bioactive chemicals using some of the same properties as Metavibola, ecological distributions, prevalence, abundance, variation across the population, and of course, association with a phenotype of interest. So just like in Metavibola, this gives us an integrated rank of not just which chemicals might be bioactive in a phenotype, but which identified chemicals they also associate with. I think for the sake of time, I'll skip over. We have an overview of the different chemical modules that we find in the HMP2 IBD dataset in particular, which tend to be chemically homogeneous and either enriched or depleted in ulcerative colitis very consistently. Some of these are modules containing known IBD-linked chemicals. Others, with equal confidence, are a little more unexpected or novel. There are things like urobilin pathway members, histidine, and pentothenate or B vitamin family members that were highly prioritized by the system as well. And again, rushing a little bit for time, in this B vitamin module in particular, most of the module members were unidentified chemicals. Only a few of them were associated specifically with ulcerative colitis. And we've been able to, with Wendy, to take at least one of these all the way through validation in notobiotic animals, looking at nicotinamide riboside as a chemical that links essentially the NAD metabolism pathway with B vitamin metabolism. So long story short, for the sake of time, in a DSS model of GI inflammation in notobiotic animals, exposure to nicotinamide riboside was able to significantly ameliorate several different measures of GI inflammation, overall histology, and this was true across different DSS models, both time courses and concentrations, such that, I gave a few examples of MetaVibula where we were able to take a couple of proteins through from a big population study to bioactivity validation, and we have one chemical so far that has gone through the same validation pathway as something that alleviates GI inflammation. So I'll wrap up there with the advertisement I promised at the beginning for a
Curtis, we
microbiome
are
center.
sorry to interrupt you, but we are out of time.
This is my last slide, so thank you.
Okay,
Um,
go ahead, but
so,
fast.
like I said, I'll wrap up there with the advertisement that I promised at the beginning for our microbiome center. Please feel free to take a look online at both any of the information, protocols, resources that are available there, or contact our acting center director, Sochi Morgan. Like I said, that's my last slide. So thanks everyone, including our collaborators on these projects.
Thank you very much, Curtis.